thanks very much for joining, everyone. My name's Amit Aggarwal. I'm the Executive Director for Medical Affairs at the ABPI, and I'm here to talk to you today about drug discovery, the journey from bench to bedside. Now, this lecture was actually delivered a few weeks ago on the 2nd of March, and you might be wondering why I'm talking to you from my living room today. Unfortunately, we had some technical difficulties on the night itself, which meant we couldn't record the lecture. So here I am today re-recording it for you today. So without further ado, let's crack on. I want to just quickly talk a little bit about the ABPI and just say a bit about who we are and what we do. So the ABPI is essentially the trade association, the trade organisation that represents innovative R&D based pharmaceutical companies in the UK. And our mission literally is to try and make the UK the best place in the world to research, develop and use new medicines. And we do represent companies of all sizes who invest in discovering medicines of the future. On that piece around wanting the UK to be the best place in the world to research and develop medicines, you'll see later on in the presentation, there are some areas in which we do really well and some areas where there perhaps is still room for improvement in terms of the UK as a whole, but we will come to that later. Just a little bit about myself. I'm a doctor by background. I trained in Cambridge and London, and then worked in the NHS for around 5 years. Was going to embark on a career in surgery before I decided to try a career change and move across to pharma. Still working as a doctor, and I started my career in Bayer. I stayed there for around 12 years in various roles, both in the UK and globally as well. And I moved to Leo Pharma as their UK and Ireland medical director, and have now been at the ABPI since December 21. In terms of what I do, so my role as medical director, we think about the whole kind of medicines value chain, beginning at R&D through to manufacturing, regulation, and all the way to prescribing and eventual use. The bit I'm, I suppose, most interested in terms of my specific role is around this prescribing and use piece. However, our conversation today really is around drug discovery, because none of this happens without this bit, without the R&D bit at the beginning, and that's really what we're here to talk about today. That's where the whole chain really begins. That's where the whole story begins. And I suppose no discussion about medicines discovery would really be complete without really talking about the start of this decade, the start of this decade, which I think it's probably fair to say had the most significant public health crisis that any of us have really ever known in our lifetimes. And it's not Will Smith with his dog in a deserted New York. The picture on the right is the reality in terms of how things actually were during lockdown, during the pandemic. You'd be hard pushed, actually, if you weren't familiar with the Will Smith film on the left, you would be hard pushed to know which was real and which was a movie set. So we've all been through a quite incredible time. And this slide you see here, this is again very early in the pandemic. This is from summer 2020, when the Duke of Cambridge, as he was then, now the Prince of Wales, visited Oxford, where one of the vaccines that became the Oxford AstraZeneca vaccine was being developed at the time. So you can see this is very much research at the bench, quite literally at the bench at that time. And when we talk about from bench to bedside, we are talking about translational research, and what we mean is that translation of scientific discovery from something that can be eventually used in patients to improve care in some ways at the bedside. So this is very much obviously at the bench stage at the moment, and this is the bedside, or the chair side in this case. This is actually my dad, who received his first dose of COVID vaccine. This was actually the BioNTech Pfizer vaccine he received in December 2020, just before Christmas. I think he was possibly one of the first 500,000 in the country or so to receive this, and has had several several more since then, as has my mum, and actually has been very effective. They're still well and fighting fit, touch wood. So that's been a really good experience for them. So we will come back to COVID later on in the conversation, but what I want to do now is just kind of really rewind back to where some of the potential origins of drug discovery are. Many of you will be familiar with this individual, James Lind. He was a British naval surgeon, a Scot actually, and this is him aboard the HMS Salisbury in 1747. So for those of you not familiar, the picture on the left describes a condition called scurvy. Now, scurvy was it was a scourge, really. It was in terms of sailors on ships, and there were many voyages where literally half the sailors died from this condition, and nobody knew why it was happening, what was causing it. And James Lind decided to do potentially, but probably one of the world's first clinical studies, possibly not the world's first, but certainly one of the first. And he had 6 pairs of sailors, and he randomized them. So to one pair he administered cider, to another pair of sailors he administered vinegar, and one pair of sailors got oranges and lemons. And lo and behold, 5 days later they were feeling better, and a week later, a week or 10 days later, they were almost cured of their scurvy. And thus was discovered the fact that, well, many years later, there was actually a vitamin C deficiency, but at that time a cure was discovered, not through medicine but through food. Although curiously, it was many more years before the use of citrus fruits became widespread on sailing voyages, and that was one of the reasons why British sailors were called limeys, because they carried a supply of limes around on vessels. Again, this is probably one of the most important health interventions, really, in health discoveries. This is the condition on the left. The young boy on the left has something called smallpox. That's a condition that actually many of us will not be familiar with at all, because it was eradicated many years ago, finally in the 1970s. Its discovery is an incredibly interesting tale. So Edward Jenner, who's a British physician and scientist, and he and I believe others, but he certainly had made the observation that in the day you had the concept, you had milkmaids. These were ladies who would milk cows and sell the milk on. And he noticed that these milkmaids generally didn't seem to be afflicted with smallpox, and smallpox was an absolutely horrible condition. It caused a lot of death, a lot of morbidity in terms of scarring and other effects, and it was a incredibly horrendous condition for those, even for those who survived it. And so this observation that these milkmaids actually didn't seem to be affected by smallpox was quite a striking one. What milkmaids were exposed to was cowpox, and cowpox seemed to be at the time a gentler form of smallpox, in that it was fairly self-limiting. The milkmaids might come down with a temperature for a few days, but actually they would recover pretty well from it. And he wondered if actually that immunity, well, the concept of immunity wasn't really known then, but he wondered if being infected with cowpox conferred some protection, if I can use that word, some protection to smallpox. So he did an experiment. With very questionable ethics by certainly modern day standards, but he, the son of his gardener, who was I believe 8 years old, made a scratch on his skin and inoculated him with the pus from a cowpox sore. And then several weeks later, again made a scratch on his skin and tried to infect him with the pus from smallpox. And lo and behold, he didn't come down with the smallpox, and thus was born the concepts of a vaccination and a vaccine to smallpox. And I think it's fair to say the eradication of smallpox is considered by many actually to be the single greatest health intervention, really, that mankind has ever known, in terms of the total eradication of a disease that had, as like I said, incredible morbidity and mortality attached to it. Not all scientific discoveries were for the good. This slide shows you 3 products: aspirin, lysitol, and heroin. Aspirin we're very familiar with and still very much used today. Heroin at the same time as aspirin was discovered, was actually marketed as a non-addictive sedative for coughs. And who knew actually just how addictive it was? But at the time, it was believed to be a non-addictive therapy. It was very effective for coughs, but as we know now, had many other pretty serious side effects. And that's what a vial of heroin would have looked like in the day when it was openly sold. Another really important discovery, the discovery of insulin. A lot of the examples we've seen so far have been British examples, and we'll come to another one. But these were a group of doctors in Toronto who discovered insulin, and in 1921. So again, if you consider what a significant condition that is and how manageable it is nowadays, and that was discovered just over 100 years ago, so incredibly significant again. And then, of course, no discussion of drug discovery would be complete without talking about this gentleman, Sir Alexander Fleming. By the time he discovered penicillin in 1928, he was already something of a scientific overachiever. He had discovered, for example, that lysozyme in saliva and mucus had a sort of a antibacterial effect. But obviously, his greatest discovery was penicillin. He had a petri dish full of growing Staphylococcus aureus on his bench, on his desk. He went on holiday to Suffolk for a few days, and when he returned, he realised his petri dish was actually contaminated with fungal spores. And he famously remarked, "That's funny," because what he saw was where the fungal spores were, and the surrounding area there was no bacterial growth. But the bacterial colonies that were far away from the fungal spores were still growing as normal. And he eventually named this mold, this fungus was named penicillin. And it was many more years actually before that was able to be purified with the help of a group of scientists from Oxford. That was able to be purified and then actually used in clinical practice. And Sir Alexander Fleming shared the Nobel Prize with those others who helped in the purification process to make a usable medicine out of it. But again, an incredibly important discovery, one that still holds as much significance today, such as the importance of antibiotics and the fact that actually we have growing resistance to antibiotics today, and that is certainly a public health problem that we need to tackle as a whole. So we've talked a little bit, we've seen some bench examples into bedside, but it actually goes, I would say, beyond that. So the bench part, firstly, can we invent a solution to a public health, or can we invent a solution to a health problem? And then the bedside part, can that invention, can that work in humans? Can that improve health in some way and be usable? But then it goes beyond that. Does it actually, it might work in a person, does it actually benefit patients? Can you deliver it reliably? Can you deliver it at scale in practice? And overall, does it improve public health? And for example, the smallpox vaccine we saw is a really clear, stark example of bench all the way to public health, but a story that's taken place over actually many hundreds of years, from the original discovery through to its final eradication in the 1970s. So we talked a little bit about medicine discovery. I mentioned on the smallpox example a little bit about medical ethics. I just want to return to this topic very briefly. This slide shows you a still from Normandy. These are the D-Day landings in June 1944. Coming up really towards the end of the Second World War, and after World War II ended, you had something called the Nuremberg trials. Now, this picture you see here, this is Karl Brandt, who was a Nazi doctor, and he's standing trial there in a famous trial called the US versus Brandt. He's standing trial for some of his crimes during the Second World War, which included unethical experimentation, forced sterilization, amongst others. and he, as well as several of his sort of fellow Nazi doctors were put on trial, and later executed for their crimes. But actually, out of that Nuremberg trial developed the Nuremberg Code. And what Nuremberg Code is, the Nuremberg Code was essentially, it forms a sort of part of the judgment of that trial, and it laid out some incredibly important principles. I mean, in many ways, it's considered by some to be kind of potentially the most important document in the history of clinical research ethics, because it's had such a massive influence on global human rights, and in many ways, it's become the prototype for other codes of ethical practice around the globe. So, without reading out the full list of 10 there, which itself is heavily abridged, but concepts such as the voluntary consent of the human subject being essential, and about the experiment should be such as to yield good for society, and should be conducted to avoid all unnecessary physical and mental suffering. So it lays out these really fundamental principles, and off the back of the Nuremberg Code, you had the Declaration of Helsinki several years later, which kind of took those principles in the code and kind of refined them further to be more kind of to be used to be more practicable, if that's the right thing to say. But this really is the underlying foundation, really, certainly as far as ethics in clinical research ethics are concerned. But more generally, in terms of global human rights and other codes of ethics, it's an incredibly important cornerstone of that. So those kind of codes and ethics and so on, they apply very much to human trials. And what you see on this slide is phase 1, 2, 3 for clinical trials, clinical i.e. in a person. Before that, you have a preclinical phase. So before medicines are tested in humans, they are tested usually in animals, and that's what we call that the preclinical phase. And really, you're looking not for the effectiveness of the drug there, but you're looking to see if there are any very obvious safety issues that arise there. And then the medicine generally moves throughout phase 1, 2, 3 before going on to regulatory review, then actually used widespread. But beyond that, there are also phase 4 trials as well. The aim of phase 1 trials, and the aims will vary according to the disease area. Generally speaking, in phase 1 studies, you're not looking to test how effective a medicine is. This is very much about safety. You're looking for obvious toxicological effects. You're looking to see how well the drug is absorbed or metabolized and excreted in the body. So when you move on to phase 2, in phase 1 is actually be done in a very small handful of patients, often single digit or low double digit numbers of patients. When you move into phase 2, however, that's where you're looking for your sort of proof of concept. Does the drug work potentially in the indication or condition that is being looked at? And those studies will tend to be a little bit bigger. As well as seeing if the drug will work, you're again always looking at safety. You're also potentially looking at what dose of the drug might be most effective, what strikes the best balance between benefit and risk. And by the time you get onto phase 3, there you are potentially studying that medicine in hundreds or sometimes even thousands, sometimes even tens of thousands of patients. And there you're pretty confident about the effect that your drug has an effect, and really you are looking to clearly define that, as well as looking at the safety profile in more detail of your medicine. It's probably fair to say many medicines will fail at any of those phases, the preclinical phase, phase 1, phase 2. By the time you get to phase 3, I believe it's around sort of a 60% chance of success. But still a high level of failure, and obviously the level of risk of failure is much higher at phase 1 and phase 2. After phase 3, medicine will go to the regulator, in the case of the UK, the MHRA, and they will review that whole dossier of evidence to determine whether the benefit risk profile of that medicine is sufficiently good to provide a license for that medicine, a marketing authorization, and then that medicine can go on to widespread use. But beyond that as well, phase 4 studies do occur. to further quantify the efficacy and safety of that medicine. So the story doesn't really end, and that's just a snapshot of some of the clinical trial phases that exist in terms of the formal bringing to market of the medicine and beyond. In terms of the sorts of clinical studies, so this kind of shows you a hierarchy of evidence. So at the top, at the top, you've got the gold standard, which is a systematic review of multiple randomized controlled trials. And at the bottom, you've got opinion or case report. So many, in terms of those phase 1, 2, and 3 studies, if we talk about phase 2 and 3, a lot of the time, most of the time, they will be randomized controlled trials. However, that's not always the case. If you consider rare diseases where you may have only a very small handful of patients, then it's often not practical to subject a very small number of patients to a randomized controlled trial. So actually, cohort studies and so on do have a place as well. And what do we mean by randomized controlled trial? What we mean by randomized controlled trial is you have a population of interest. So if you think of if we're doing a study in let's say diabetes, all the patients who have diabetes and which is your population of interest, and within that we will pick out a sample population, so a subset of that population who have diabetes, and we'll test our therapy. Treatment A, say that's the drug or the investigational medicine where we're looking at. We will compare that with treatment B. Treatment B might be current standard of care, so what's currently being used. It may even be placebo, placebo in terms of a dummy medication or something that is expected to cause no treatment effect. And what you will often do in a randomized controlled trial is randomize your sample population to either the investigational treatment or the standard of care placebo. It's probably worth just talking about the placebo effects, because there's a temptation to think that placebo is entirely without effect and is always you're always getting to the nub of the treatment effects with your investigational medicine when you're comparing with placebo. Not always the case. This slide is a study in migraine patients, and what you have here is you have 3 sets of migraine patients. So you have a set who is randomized to no treatment whatsoever, so they received absolutely nothing. You have another group within that who was randomized to receive a placebo pill, and then you had another group within that who was randomized to receive something called Maxalt, which was the migraine medicine being tested. Now, the interesting part here is how those patients who received the placebo pill or received the Maxalt pill, how their pills were labelled. For those receiving a placebo, they were either labelled as placebo, or they were labelled as unspecified, or they were labelled as Maxalt. So this means a patient receiving a placebo pill would have had a pill labelled actually as being the actual medicine. So they would have thought potentially that they were receiving actual medicine. Of the patients receiving the actual medicine, Maxalt, these people here had their pill labelled as placebo, unspecified, or labelled in truth as Maxalt. And what you see here is really striking, actually, because you look at the difference in treatment effects between those who received a placebo pill labelled as medicine and those who received a medicine pill labelled as placebo. And you can see it's actually not a huge treatment percentage change in the pain score there. It's actually the treatment effect is actually fairly similar between the two. And to compare with, this is again the patients who received no treatment whatsoever, and you can see the massive delta between hair and hair. So I think that speaks to the potential power of the placebo effects, and it's often quite striking in for example pain studies and the like. So just a word of caution when it comes to placebo, there's potentially more to it, just more than just positive thinking. Just switching gear very quickly, we talked about medical ethics, we've talked a bit about trial design. that's not always enough. And we talked a few slides ago about this regulatory regulatory review at the end of phase 3, and I think this example here underlies why regulation is so important in the whole journey from bench to bedside. This remarkable individual here is Frances Oldham Kelsey, and she was a reviewer at the US regulator, the Food and Drug Administration. And this story takes place in the 60s. So the lady you see on the left who's feeding her child, you can see she has a missing arm there, and she's feeding her baby with using her mouth to hold the spoon. She's someone who'd been exposed to thalidomide when she herself was a growing baby, was in the uterus. She was exposed to thalidomide, and as a result had birth defects, missing arms in her case. And thalidomide affected scores of mothers and children in Europe and in Canada. And many of those individuals who were exposed to thalidomide while they were in utero are still alive today and living with the consequences. The US, however, was less affected by this, and it's largely due to the efforts of this, as I said, of this remarkable lady, Frances Oldham Kelsey. Because although thalidomide had previously been approved in Canada and more than 20 European and African countries, she withheld approval for the medicine. She requested further studies. Specifically, she had noticed an English study that documented something called peripheral neuritis, sort of a nervous system side effect, and she requested more information on that. And she also wanted data to prove that the medicine wasn't harmful to a growing baby, because what I haven't mentioned to you so far is that this drug was actually prescribed for morning sickness. So it was prescribed specifically to help alleviate the symptoms of morning sickness. So obviously any growing fetus would be exposed to that, and she requested specifically data that could show the drug wasn't harmful, which sounds very sensible and logical and obvious. But she was the one that did that, and that data was not forthcoming to her satisfaction, and so that medicine was never approved. And she really a hero in public health terms, and was awarded various honours. And she worked into her early 90s and then was over 100 years old when she eventually passed away a few years ago. So quite remarkable story in the case. So what we've done, we've really focused so far on clinical trial phases, and in terms of human trials, essentially. But there's a whole story before a medicine actually gets into humans, or even animals for that matter, in terms of your preclinical studies. Where does that medicine to test actually come from? If you're not Alexander Fleming, who's come back from holiday and discovered that a fungus has accidentally grown on the Petri dish, if you haven't accidentally discovered a medicine in that way, where do you get your medicines, your medicine candidates to test eventually in humans? And the answer to that is in this area here, where we talk about lead discovery, lead optimization, and earlier than that, target validation. So what do we mean by that? What we mean is how we discover drug targets today, and this kind of just lays out essentially five steps in that process. The first process is one of target identification. So this is the initial stage of discovery, where researchers, working in a pharmaceutical company or in academia and universities, where they identify a specific biological target that they think might be associated with a disease or a condition. So the target might be a protein or an enzyme, a receptor, or some other molecule that plays a role in the disease process. And then once you've discovered a target, researchers search for compounds that can interact with that target and potentially inhibit or modulate or potentiate its activity. So you're looking for hits. It's a bit like bit like imagining a lock and trying lots and lots of keys. Some will not fit at all. Some will fit. Others will turn or partially turn that lock. So in a way, your lock is your target, and your key are your 100 keys you're trying till one opens a door are your hits. So once you've identified some hits, some keys that might fit in that lock, researchers optimize those hits to improve how potent they are, how selective they are, and other properties that make them better as drug candidates. Then they further optimize those leads all the way through to preclinical development, where those selected drug candidates, and there will be very, very few from the original hit discoveries, are looked at in animal models to determine their safety, how well they're absorbed and metabolized, eventually through to clinical trials in humans. So one of the key aspects here is that hit discovery. Now, how is that done? This is something called high throughput screening. So this is one process that's been used for a number of decades now by pharmaceutical companies, and essentially they have libraries, tens of thousands, hundreds of thousands, even millions of compounds that will be potentially filled up in this pipette and will be tested against some of those targets, whether it's a receptor or protein or an enzyme, will be tested. And so these are fully automated labs, and they will be testing many hundreds, thousands, tens of thousands of compounds, meaning potential medicines, on these potential targets to see what do you get a hit. You get something that works in that petri dish environment, in that assay. So that's the process known as high throughput screening. So high is in a high volume, and you're putting that through a screening process very rapidly and very quickly and very effectively. This slide just shows you a bit more about that process. So you have your screening library of compounds which go through high throughput screening, and for any that may have drug-like properties, because some will have no drug-like properties and some will, depending on your target, you try and optimize that eventually into clinical trials. And if you are very, very, very lucky, through to final regulatory approval, FDA in the USA, but it would be MHRA in the UK here. And here are some examples of how that approach actually works. Well, yes, actually, here are some examples of where at the top, imatinib, which is a targeted therapy used to treat a type of leukemia, was discovered using this approach. This antiviral to treat flu, an anticoagulant medication used to prevent blood clots, which inhibited the activity of something called factor 10A, which is essential in the clotting cascade, and an antidepressant. So all of these 4 medicines were discovered using that high throughput screening approach, which is actually quite remarkable. And in terms of knowing where to look, where should companies focus their where do companies focus their efforts on? How do they decide where to focus their R&D efforts? This is a survey done of scientists in pharmaceutical companies from not very long ago, actually. And you can see, in terms of very important, you can see that as well as multidisciplinary collaboration, access to technology and data, which kind of goes without saying, but it's actually that medical need, that unmet medical need, that societal need, which is rated as very important, or important to the vast majority. So that reassuring to see that really is the driver of where companies try and focus their R&D efforts. I wanted to quickly return to the topic of COVID, but before COVID, just really kind of highlight the role that women play now at the forefront of translational research. Many of those examples you saw earlier on in the presentation were very kind of male heavy, with your kind of Edward Jenners and Alexander Fleming, but actually now women very much at the forefront of this. And on the left, you've got Sarah Gilbert, who led the team who discovered the Oxford AstraZeneca vaccine. And on the right here, you have this gorgeous husband and wife couple, so Dr. Özlem Türeci and her husband Ugur Sahin, who founded BioNTech, and they obviously developed the BioNTech Pfizer vaccine. And they, even went to the lab on their wedding day many decades ago. And I particularly love this quote from Sarah Gilbert, who in an interview with the Times, she said she had an email from a parent of an 8-year-old girl who said that she'd seen her on television and didn't realise that women could be scientists, and now she wants to think about being a scientist. And Sarah Gilbert's absolutely brilliant response was, well, what on earth have you been teaching her? If your daughter didn't realise that women could be scientists, what on earth have you been teaching her, for heaven's sake? Which I think is a fantastic, fantastic quote, really, and sums things up pretty nicely. On the topic of the vaccines, the COVID vaccines, the two vaccines developed, the Oxford AstraZeneca vaccine and the BioNTech Pfizer vaccine. Really interesting, because they were using technologies that hadn't, certainly in terms of mRNA, in the case of the BioNTech Pfizer vaccine, hadn't been used before, and the adenovirus vector used by the Oxford group also hadn't been used much before, but both fundamentally doing very, very similar things. In the case of those vaccines, both vaccines, one by introducing a strand of messenger RNA, one by introducing viral DNA using a viral vector, both utilizing the cell machinery of a person to generate the spike proteins that would be recognized by a person's immune system and combat the virus early on after someone's been infected. So whereas many vaccines, traditional vaccines, if you think of the smallpox example, introduced the actual antigens themselves, what both of these vaccines were doing was actually giving human cells the means to produce that antigen themselves that could then be recognized by the immune system. So different mechanisms, but essentially achieving the same outcomes. And if we talk on the subject of vaccines, there is a huge amount going on in the vaccine space now. And actually, if we take COVID vaccines out of the equation, actually over half of the vaccines in the pipeline are targeting diseases for which there's currently no vaccine, which is absolutely fantastic. And that's 71 vaccine candidates in the offing at the moment for some quite significant conditions. Clostridium difficile, which is a horrible infection that causes life-threatening diarrhea, particularly in elderly hospitalized patients, and great to see a vaccine potentially being developed for that, as well as Lyme conditions such as Lyme disease. And what's quite interesting about this slide is looking at the vaccine technology. So you can see, as well as the viral vector platform used by the Oxford group, which is quite prominent, you can see the mRNA platform used by the BioNTech group and now other companies as well. You can see how that has become so important in terms of the vaccine pipeline, and striking to think that pre-2020 that had never been used in a vaccine before. So it shows you how quickly this technology is advancing and the potential for good it has moving forward. And as we're on the topic of mRNA and DNA, and I don't think any discussion around drug discovery would be complete without talking about the role of genomics and the discovery of the genetic code. This is Francis Crick, James Watson on the left, and Rosalind Franklin and Maurice Wilkins in the middle and right, and their discovery of the double helix structure of DNA. And this led in the 1990s to something called the Human Genome Project, and this was a multi-country project to map the entire human genome, well, certainly over 90% of it, and one of the rare examples of a project that came in ahead of schedule and under budget. And it's, I think, after the Human Genome Project was essentially complete, I think there was an expectation that there would be essentially kind of a rush of medicines based on our new knowledge of the human genome. The reality is it's actually taken a bit longer than we thought, but is now starting to bear fruit, as we'll see in a few slides' time. This is the cell and gene therapy Catapult. So this was set up with the help of government funding in 2012, initially at Guy's Hospital. And This cell and gene therapy Catapult was essentially designed to help small startups, biotechs with that research infrastructure, that manufacturing infrastructure, as well as expertise with regards to regulation and access, to help those small companies bring their cell and gene therapies to patients. Because as I said, although the Human Genome Project ended in 2003, it was actually a lot more work needed to be done to actually translate that into medicines that might actually be usable. And so there's a recognition of this burgeoning technology around a decade ago, and thus was born the Cell and Gene Therapy Catapult. And in the space of cell and gene therapies, there's some incredible, quite remarkable things happening now. This is a therapy called CAR T therapy. So this is a type of treatment in which a patient's T cells, a type of immune cell, they're changed in the laboratory so that they will bind to cancer cells and kill them. So what happens here is blood from a vein in the patient's arm is taken, it flows through a tube to something called an apheresis machine, and that removes the white blood cells, including the T cells, and then sends the rest of the blood back to the patient. And then the gene for a special receptor called a CAR, chimeric antigen receptor, that's inserted into the T cells, and then millions of those CAR T cells, as they're called, are grown in the lab and then given back to the patient by an infusion. And those CAR T cells, they're able to bind to an antigen on the cancer cells and kill them. So an incredibly involved process, but actually achieving a cure for some of these patients with types of lymphoma, for example. So really quite remarkable. This is a young lad with a condition called spinal muscular atrophy. So this is a rare genetic condition, and it affects a gene called SMN1, so survival of the motor neuron gene, and there are also other kind of rarer versions of that as well. But this faulty gene essentially means that the nerves in the spine that are carrying messages from the brain to the muscles, they become injured over time. They die off. And so these individuals, and usually children, they become weaker and weaker, find it hard to move, hard to breathe, and eventually die from it. And what you have now is you have a medicine available that's a gene therapy. It uses a virus, a viral vector, to insert the missing SM1 gene into a child's body, and that can actually stop the gradual muscle weakness happening if given early enough. This is a type of gene therapy that is being used today, and again, absolutely remarkable for a condition where previously there was very little hope. These two siblings are Nala and Teddy. Nala has a condition called MLD, and unfortunately treatment for her wasn't discovered in time. But because she was known to have this condition, her sibling Teddy was able to get treatment. What is MLD? MLD, again, caused by a faulty gene, and it means that children can't produce an important enzyme called ARSA. This enzyme, it helps, a deficiency of this enzyme basically means that fatty chemicals called sulfatides, they build up, and what they do is they gradually destroy the protective myelin layer, the protective fatty layer around cells in the brain and nervous systems. So essentially it's a demyelinating disease, and again you get this devastating deterioration where children lose the ability to walk or talk or eat, and eventually to see or hear. And what the therapy now available here involves hooking up, in this case Teddy, hooking her up to a machine where her blood is removed and filtered so that single bag of stem cells can be collected, bit like dialysis, and then those cells are then exposed to a viral vector which inserts a working version of the faulty gene, and then those gene-corrected stem cells are then infused back into Teddy to replace, and she's also given chemotherapy to destroy the faulty stem cells in her bone marrow, and then these corrected stem cells are infused back, and again a potential cure for her and has been used in other children as well. So really quite remarkable. So in terms of what's making its way to the bedside, this graph just shows you it's a big focus on oncology, but also other conditions such as CNS, a lot of activity happening in the Alzheimer's space, metabolic diseases as well. So it'll be really interesting looking forward to see what comes over the next 5 to 10 years. How much do these medicines cost to develop? So what this is like, this is an analysis from Deloitte from last year. And what this essentially shows you is the average cost of medicine, of drug discovery from discovery to launch, creeping up gradually over the years. The time taken to develop also going up. You can see from sort of 6 years to 7 over the last nearly the last decade, partly as a result of the increasing complexity of studies. You saw some of the examples just now about actually how complicated some of the science involved is, but also difficulties in recruiting patients as well. This looks at the return on the late stage pipeline. So you can see the returns generally have been dwindling. Now you saw this kind of uptick in 21, largely driven through emergency use authorizations from COVID. There's another graph actually. If you strip out those emergency use authorizations, so those COVID medicines, that would actually sit at 2.9, and then that would sit at 0.6, thus continuing that kind of downward trend you see. So drug discovery is becoming more expensive, is taking longer, with potentially lower returns, which is something to bear in mind. Because actually, if we look at the UK itself for a moment, this is an article from The Guardian from a few weeks ago. And what this warns is that in terms of that whole landscape around medicines R&D and drug discovery, we are really losing out. Whereas we were once a leader in this field, that is potentially not the case anymore. This shows you the number of industry, so commercial clinical trials initiated in the UK year on year. And what you see there is a massive, over sort of 40% drop between 2017 and 2021, which is a worrying trend. This table on the left, this shows you where we ranked. So this shows you phase 1, 2, and 3 studies. If you look in 2017, we were number 4 in the world, behind only USA, Germany, and Spain. Pretty remarkable, punching well above our weight. Today we languish at 10th in the world for phase 3 clinical trials, and we've fallen down the ranking for phase 1 and 2 as well. So really quite worrying. And it's worrying for a multitude of reasons, because actually clinical trials, performing them in the UK, they really matter. Clinical trials, for the most part, they are address an unmet medical need. If you're in a study, it will more often than not mean you're in the study because there isn't a medicine available for you that currently works. So clinical studies going elsewhere mean that the UK patients miss out on that. Making sure that our healthcare professionals, our doctors, our pharmacists, our nurses get experience in using medicines in clinical trials is really important to drive uptake of those same medicines after they're approved. Because if they don't have experience in clinical trials, and actually the chance of uptake of those medicines becomes lower after those medicines are formally made available. And if we aren't using medicines to the same level as other European countries are, then our standards of care have the potential to be lower than elsewhere in the world. Not always, but they have the potential to be, and that might well affect future clinical trial placement in the UK. It's also worth bearing in mind that these medicines in clinical trials, they are free. They're free to patients and they're free to the NHS. And actually, clinical trials are a net revenue generator for the NHS, an average £9,000 per patient. So in theory, it should be self-sustaining. And actually, clinical trials more generally, they generate growth in the economy. And one estimate, actually our ABPI estimate, puts this as at 68 billion in additional GDP, gross domestic product, over the next 30 years, if our R&D spending rose to be on par with the US. And we're really, really good at life sciences. You saw the examples, the historical examples, and the more recent ones, such as the Oxford AstraZeneca vaccine. We're really, really good at this. We excel. So it's incredibly important we don't squander that opportunity and talent that we have in the UK. And if we're going to be successful, it needs to be a collaborative approach between industry and academia and the government and the MHRA and the NHS. This is incredibly important. On my penultimate slide, I think really important to just very briefly talk about the potential for AI. AI is with the advent of ChatGPT and GPT-4 and Google Bard coming along. AI is I think shocked many with its potential, and I think the potential for its use in clinical trials and drug discovery, whether it's speeding up drug discovery, helping identify targets or other genetic and biological factors that might contribute to someone's response to a drug, improving trial design, enhancing safety, discovering new purposes for medicines, drug repurposing, identifying existing drugs that might be effective for new indications. And in silico trials, this is where you can essentially, the potential for doing virtual clinical trials, computer simulations of clinical trials that can be used to evaluate safety and efficacy, meaning you may need to do less animal studies or less human studies. The potential is huge. There's already chatter around how AI might be helping identify medicine targets or refining some of the medicines we already have. So watch this space. It's going to be a really exciting next 5 to 10 years. So in summary, I can't emphasize enough how important translational research is, bench to bedside. It really, really matters. And it's had a unfathomable impact on health, both from an individual level, but as well as public health and the societal perspective as well. The basic science and the commercial studies are both essential. They're symbiotic, and that bench to bedside only works when both of those happen. And it really does need a multi-stakeholder approach for success. As you saw, genomics is starting to bear some fruit in therapy and medicinal terms, in personalized medicines and the potential for one-shot cures. Those examples you saw towards the end, those are all potential cures administered once, and potentially nothing needed ever again if that condition is caught early enough. So the potential is absolutely huge, but medicines development is an increasingly expensive, low return proposition. So only the best countries will win the race to conduct this research. And we're in a bit of a hole right now as far as a big segment of translational research is concerned, commercial clinical trials. But we can dig ourselves out of this because we have the potential to do so. So we are, in summary, we're typically great at this. And if you're interested in getting involved in translational research, there are incredible careers to be had, discoveries to be made in this space. So if you're interested, please ask, seek out more information, and very best of luck to you if that's the path you choose to go down. Thank you.